Thanks for joining me. My name is Jerry Skeffis, and I'm with the Metacell team. We worked during the Codeathon to develop a tool to integrate two different pieces of software so that you can visualize neuronal networks from SCAN in the Open Physiology Viewer. And so what does that mean? SCAN is kind of the pathway at the bottom here where you're taking models that have started as NLP models, and then you've got expert curators that are working to refine those neural network models and put them into a database. But then app anatomy is a methodology for expert curators to create a circuit diagram showing all of the different connections throughout anatomy so they can represent physiology in an anatomical context. And we thought it would be really cool if you could take these models, all of the models coming from this SPARC connectivity knowledge base, and represent them in app anatomy so you could learn a lot more and explore how they work. So this shows the tool Scan Composer that we've previously been working on, and we have ongoing projects to develop. And then this makes very simple point A to point B network diagrams, and you can actually expand those over time as we continue to develop the tool. App anatomy, on the other hand, has been going on for quite a while and has a lot of different complexity that you can add in there. So you can add multiple scales of anatomy coming from organs down to tissues and cells and subcellular locations, and the same schematic can be used across all sorts of different species and across all sorts of different physiological scenarios. So the approach for this project was to take the latest release of the database coming from SCAN, and that comes out as a turtle file that then needed to be converted to a JSON and in turn converted to a spreadsheet, which is what the Open Physiology Viewer can use. So it's a little more friendly for people to create new models. And the asterisks on this diagram show the different components that we worked on for this project. So basically, all of the models that were given to us are able to be imported into the Open Physiology Viewer. And I'll show you a quick little video of what that looks like. These are some of the models being brought in, and you can also see that you're able to see it in the context of the whole homunculus of all the different organs. It shows kind of spleen and thymus and things like that. So I'm just going to proceed. This shows also the extension of a model to show a comparison between sexually dimorphic development, because a lot of the models that were given were about the reproductive system. So we showed the female versus the male in pink and blue. And then we provide links to some additional models that were provided. So this is our team. Please reach out with any questions, and we look forward to continue to work on these projects. Have a great day.
Hello everyone, in the SPARC Codeathon, we present Scan Compare, a Python package and web app to visualize scan data across different species. Now, what is the project motivation? Before that, let's go to what is Scan, an established semantic store containing a variety of information. Why Scan? Because it follows the principles of FAIR. How is Scan organized? It is basically in the form of connections. Now, what is main motivation? We want to make it easily visualize it, so that user can easily visualize everything. So it's always about easy to understand visualization. Now, specific project goals is to retrieve anatomical data, map it to different species, different phenotypes, and then finally visualize it for user to access it. Now, how did we retrieve anatomical data? We set endpoint at place graph, use SPARQL and Python to filter out all the information. Now, to visualize the anatomical data, we manually mapped the basic anatomy of 3 different species, manually assigned different regions to those in the regions in the scan data, and then finally used Cytoscape and Plotly to plot the connectivity data frames. Now, we have 2 different platforms. First one is Compare Python package, and second one is web application. In Python package, it is customizable, portable, and easy integration is there. It is also reusable, whereas web application, it is easy to use, quick visualization, and does not require computational experience. Here is the demonstration of Python package. Here you can see there are different neuronal connectivity for different phenotypes. You can go to any region and it will get you the label of that region. You can go ahead and zoom and you will be able to see that zoomed in area, and you can again go to different areas and have a label of that particular area. We have also a simple representation of selected regions. You can go and select in the Python package the specific region and it will plot the simple representation. We also have projection fibers, which basically says that neuron A project to neuron B. Similarly, on web app, you can choose different species, different regions, and have a simpler representation. You can also have a complex representation very similar to a Python package. Now, what does this mean for the user? It allows communities who have limited technical abilities and who do not know a lot about anatomy to go and visualize different models. Now, in future, we want to integrate this with Spark Flat Map. We want to include different species and phenotypes. We also want to include app anatomy, functional connectivity models. Finally, we would like to thank Spark and Scan team. Thank you so much for providing us this with opportunity.
Yep.
Get started. Hi, our team represents the Project AI SPARC Flows, a tool for fast and small savable workflows for SPARC data analysis. The team includes me, Charles Horn, John Bentley, and Mason Means. AI SPARC Flows is a dashboard that allows users to input a desired analysis workflow performed on SPARC datasets to do machine learning and descriptive analytics. The requested analysis is used to generate a prompt that is sent to a large language model. In this case, ChatGPT using the OpenAI API, which in turn generates a Docker file and a Python script file that is returned to the user. The user runs the Docker file to create a Docker image with an appropriate analysis environment and built-in Python script to complete the analysis. So why use this tool? There are two pain points that we deal with for analyzing SPARC data. One of which is fast preliminary analysis, and the second one is to create small footprints of saved workflows, both of which align with our goals and benefits to the SPARC community. Second and thirdly, I want to mention that our approach is lightweight, dashboard implemented in the cloud or locally for the creation of workflow files, including this Python script and the Docker file. And we see this as highly feasible in the sense that we have lightweight files implemented quickly in the cloud, and we also can train the model again on large language models that might be open source in the future.
The dashboard is implemented with a Flask backend and a React frontend. To install it, simply clone the Git repository, then navigate to the dashboard directory, then run npm install and npm start. For the backend, navigate to the backend directory. Make sure you have the Python library prerequisites installed through pip, including Flask, OpenAI, TikToken, and PyYAML. Then start the server by running python server.py. Make sure you have your Open API key in the directory above the GitHub repository. Set up your local directory structure by having a subdirectory named results and another subdirectory named data. Then go to the SPARC science repository, select your data and download it to the data directory. Make sure the data is in an appropriate format, such as CSV, Excel, or XML. With further development, AI can be used to format the data.
This is an image of our dashboard. We have 4 inputs that we feed to our backend, which then generates a prompt that we feed to our model. Those 4 inputs are the analysis type that we want to run, the file type of the data that we run the analysis on, the included Python packages that will run in the container, and a short data description of the data set that we will be running our analysis on. Once these inputs have been placed, the user will click the generate button, which will then generate the Python script and the Docker file that the user will then download by clicking the 2 download buttons on the bottom of the screen. Once the files are downloaded, the user will place them into a directory similar to the structure that John mentioned previously. And then navigate to that directory, build the Docker image, and then run the image, and then run the container to perform the analysis. The analysis will then, and other generated files will then be placed into that directory, according to similar to the image seen in the bottom right-hand corner. The output and results will be sent to the results.txt file. In the future, we're very excited for the directions that we can take this model. Obviously, we can aggregate multiple data sets into one. We can use open open source LLMs instead of the OpenAI, which is not open source, and hopefully train those. Obviously, placing this on the cloud is very exciting, to be able to alleviate hardware resources constraints and potentially difficulties downloading and using this tool locally. And hopefully we'll be able to place this in a place that has high availability and will increase the ability for users to access and utilize the amazing data sets that SPARC has provided to the public. Thank you very much.
Welcome to our SPARC Flow presentation. The SPARC project provides well-curated datasets for advancing bioelectronic medicine. And what researchers can do is process this data in workflows to generate novel results, insights, and knowledge. However, there's currently no option for users of SPARC data to easily describe their data processing workflows and their tools in a fair manner. It's also difficult for them to run these workflows and reproduce their workflow results. So our solution was to develop SPARC Flow, a Python tool to describe and run tools and workflows for processing SPARC datasets in accordance with FAIR principles. We created a series of tutorials to help highlight the features of SPARC Flow. This includes showing how we can reproduce a data processing workflow from an existing SPARC dataset. And we show how we can describe this workflow using standardized workflow and tool descriptions. And we then show how we can run this workflow both locally and on cloud-based platforms. We also proposed guidelines for enabling fair workflows and show how we incorporate these best practices in these tutorials. In terms of impact, SPARC Flow supports the SPARC DSC and community developments. For example, SPARC Flow could be used to describe tools and workflows that can then be contributed to the SPARC portal. SPARC Flow also allows workflows that use SPARC data to run on different platforms. And SPARC Flow also supports our vision for enabling tool and workflow descriptions to be included in knowledge bases such that we can ultimately look towards automatically assembling workflows for different applications. Thank you from the SPARC Flow team.